I will get things started. We're continuing our celebrity showcase of nutrition researchers from animal biosciences, and this week we're really happy to have Dr. Kiarie and his lab group present things that they're working on. And Elijah has been in our department just for a short 5 years. Before that, he was working with DuPont, and before that he was researching out in Manitoba, so we may have some visitors from out west for this event. Elijah holds the McIntosh Family Professorship in Poultry Nutrition here, and I'm really going to let him give an overview of what the research is that they're doing, but he also teaches our fourth-year poultry nutrition course to the undergrads, and so there's a lot of influence going out into the industry from that. He's also teaching part of the animal disorders class, and I know he's involved in the growth and metabolism course for graduate students. So he's got a good teaching overview, and he's going to show his research to us today along with some of the PhD and postdocs that are in his lab. So Elijah, we look forward to your talk.
Okay, yeah, thank you, John, very much. First of all, I want to actually thank you and Jennifer, Dr. Jennifer Harris, for seeing the need for, reviving the Center of Nutrition Modeling seminars, at least to give an opportunity for many nutritionists we have in the department to share with the department as well as to the community the kind of stuff we do. They are cool stuff. We are actually solving many problems for producers. And today I'm really happy to share with you as well as many people who have joined this call. It's amazing. Some of the things we are doing in the front of poultry nutrition. And the time is short, obviously. We cannot be able to tell you everything we do. But I really thought that we can focus on showing you, some of these new areas we have started investigating, trying to integrate some aspect of poultry breeder nutrition, and of course, to improve the productivity and robustness in chicken. When I decided to become a professor, I was actually told that students are the ones who are the smart. In other words, the underlying smartness is with the students. And for that reason, what I will be talking about here is really student work. And most of these students you speak to you, tell you what they are up to. And I'm really pleased they are here. Dr. Reza Kibari Mohadem Kaki graduated a couple of months ago. He is now working in trial nutrition. We also have Izariwa, we normally call her Ash, the Nabaran. She's a current PhD student. We also have Rose Quito. She is actually a student of my colleague, Dr. Tina Widuski. We collaborate with her. I will explain how that's happening. And of course, a visiting student from China, Dr. Zhuang Lu, who visited with us a couple of years ago. And of course, also did some work in this theme. But before I start explaining to you what I do, I think it's very important to remind many of us who may not know that the reason why we are here today is because of this gentleman here, Jim McIntosh and Brenda McIntosh family. This family, which come from Huron County in Ontario, in a small town called Egmontfield. They saw the need to establish a poultry nutrition position 8 years ago in this department. And I have to tell you, I always cherish visiting with Jim. I've almost made it a tradition to hang out with them every summer. And of course, occasionally I do take the students, these are the future generation of poultry nutritionists, to hang out with Jim, if not for anything, to just say thank you. And of course, get a word or two of wisdom from Jim. Jim was a master student way many years back in this department, did his master's in poultry nutrition, went to Huron County, established a very successful egg production farm, and he is retired. And when he was retiring, he decided to create this position. I guess personally, I've learned a lot from Jim, and I continue learning from him, both personally and professionally, and I also always cherish, hanging out with him. I think from my lab and the students who have left already, and those who are in the lab, ours is to wish Jim and Brenda happy retirement, long life, healthy life, and all fun they can do as they travel around North America when the situation allows. But what do we exactly do? I think it's always very important to reflect on history to understand, for example, what make me stay awake at night. And I think I want to take you back in 2004. In 2004, I don't know many of you remember what was happening during that year, but that year I had just graduated with a master's degree, and I had got this cool job in the industry as an animal nutritionist. And I thought I made it, all right? And one morning in the spring of 2004, the boss came in the office and told me, look, we have a new account for broiler farmer, and he wants us to make a broiler feeding program to have this chicken hit 2 kilos or heavier in under 40 days. But here is the deal: we don't have corn, and if we find any corn or cereal grain for that matter, it is too expensive to actually feed chicken on it. So if you have to make this program, you have to use co-products or byproduct from milling and all those kind of feed stuff. And I remember sitting on my desk and asking, how is this possible? And I think to cut it short, that particular question led me to, in a couple or few months down the road, to what I was called the cold capital of Canada, Winnipeg, Manitoba, to basically start my PhD, where I actually looked at aspect of using feed enzyme to improve utilization of byproducts for feeding chicken and monogastric in general. But as a person who actually have a lot of interest in agribusiness, I make it a point to read news published in this kind of magazine like Feed Stuff. And I still have this cutting I read in 2004, where the pundits had asked this fundamental question: will there be enough corn to supply future needs? And I think they were actually talking of data from FAO, Food and Agriculture Organization of United Nations, where they are predicting that increasingly the amount of corn being produced between one harvest and another harvest is actually being consumed, and increasingly we have less and less corn in the stock for future needs, for any emergency needs. And this basically means to pass on in the feed industry that the feed prices are very unpredictable, and in most cases they always swing upward. Those who know what has been happening the last 1 year, the feed prices have really gone up. I think I was reading some report in some market, corn has gone up by 100%. But what does this mean to a poultry producer? I think whenever I have conversation with students, and I normally tell them, my research address producer pain. So if you cannot look at your research you're doing with me and see the pain you're addressing, which is relatable to a producer, maybe you're in the wrong program, to be honest. So you see, the farmers are in the business of growing chicken to make money. It has to be profitable. If it's not profitable, they are not in that business anymore. And if you look back before 2009, the feed prices were actually really affordable, and the farmers would make more money. I think the margin over the feed cost was really good. But what has been happening since 2009, you can see, is almost certain that whether a farmer make profit or not, it depends on the feed price. And you can see here that other than just some small break they had around 2014, 2019 there, the prediction does not look good either. The feed is costly. So the big question is, us here doing research, training the future generation of poultry nutritionists, what can we do? I think the first thing we start doing is getting more from what we have. And it's kind of amazing and interesting whenever you interact with students and ask them, when you see chicken poo, what do you think of it? Well, most of them say it's gross, it's smelly. But I do let them know and understand that actually that chicken poo tell you that there's a lost opportunity. It's money in the drain, basically. And it also tells you that there's a problem in the gut of the chicken, that they cannot digest what we feed them. And if you actually try to address a pain being experienced by producer, we need to get more from the feed. And in a typical diet, we feed broiler chicken here in Ontario, the corn soybean meal-based diet. You find that about 15% of corn, of protein and fat is not digested, and about 3% of starch. Now, if you apply stoichiochemistry, that will tell you is about 305 kilocalories undigested. Now, if you talk to any nutritionists and tell them, if you can salvage just 50 kilocalories of this energy, which the body will never recover. And the second point is that these nutrients, once they leave the small intestine, they actually go and fuel pathogens, which cause a lot of problem. A 50 kilocalories, not even 100 kilocalories, salvage of this energy will mean a lot in the bottom line. And basically, we spend a lot of time on a key focus in this program. Again, going back to the Jim, Brenda, and Macintosh vision, to actually be able to help digestion, improve digestive tract. Look at the new feed enzyme. Believe me, feed enzymes, we have used them for more than 50 years, but there's still a lot we can do with them, I believe. They are new molecules, more efficacious. They are also new application concepts we are trying. We also look at the feed processing. I think there's a lot of anti-nutritional factors still in many feed ingredients, which actually cause all this problem of indigestibility. And of course, chicken, many of you may not know, do not have teeth, and their teeth is gizzard. So one of the things we can do, and we actually see significant benefits, is actually manipulate the particle size to actually improve something like nitrogen digestibility. We also try to look at the novel and emerging feed stuff. So when it comes to addressing nutrition, to me, this is fundamental to a nutritionist. You need to be able to drive the delivery of the nutrients you are feeding the chicken to drive growth or egg production. But really, farm realities are different. And if you look at the genetic potential of the chicken, the genetics tell us, and actually what you see in the farm, there's a gap. And this gap is driven by many factors happening at the farm. Okay? Other than digestion or the poor feed quality, you have other aspects driving this gap. So not only that, our producers are under a lot of pressure to actually stop practices they have known for decades, if not for centuries. For example, the use of antibiotics for growth promotion. Okay? And obviously, not only that, you start seeing challenges with dysfunctional guts. We look at the young chicks and young, they'll have immature gut. That gut is really highly susceptible to enteric diseases. You also have numerous metabolic disorders. You talk of broilers, you have all leg problems, lameness with the breasts, and laying hens, we have osteoporosis, shell quality. So what we have done is try to address this addressable gap. It's a clear addressable gap. How do we close this gap? The way we look at the research in our group is we now don't ask who came first, the egg or the chicken. We know the egg came first. So you have to have that egg with the right nutrients, with the right factors before it's hatched, because a good egg will give you a good chick, and a good chick will be able to withstand all those challenges we talked when it comes to, producing the meat for broilers or turkeys, and as well as the egg production. So we have, in the last few years, actually spent a lot of time trying to understand how does broiler breeder nutrition actually influences the kind of chick you get, and not only that, how does that influence the long-term performance of that chick, the metabolic functioning, the aspect of gut health, skeletal development, and those kind of factors. We also trying to do some work in novel nutrition, but as things stand right now, novel nutrition is still academic concept, not yet to be commercialized. And as well as we look at the early life, how can you make these chicks actually have a good start? Can we have better feed ingredient processed properly, meeting the nutrient requirement for the moment? At that moment, the chick need to grow the gut. Can we grow the gut? and make sure that that chicken in 10 days or so will be able to withstand, dealing with more feed ingredients. Okay. So one of the concepts I've tried to address is this breeder nutrition, and I actually share this with my colleagues, Dr. Hubert, Dr. Steele, and Dr. Wood. They do work with species which are mammalian, and we normally joke they are really different from chicken. But one unique thing about chicken is that we have separation of dam and the embryo during development, and that's presents a very unique opportunity. But there's a lot of work in human actually showing that if you actually manipulate the broiler breeder nutrition or the dam nutrition, you actually influences much more than the quality of the hatchling or the newborn, but also their long life, and also whether they are able to withstand environmental stresses and any other stresses they will have. And of course, very important, the influence, the structure and function of tissues and organs, which is very critical for us in broiler production. But this is not new. I think the industry practitioners know that the chick quality can actually be influenced by what we do in the broiler farm, broiler breeder farm, what we do in the hatchery, what we do when you're transporting the chick in the barn. But what we are really interested is to understand whether these factors do influence the performance, do address some of the challenges we see in these birds, do influence the metabolic functioning. And our initial focus is on these functional ingredients or nutrients, omega-3 fatty acid and the yeast metabolite. And before I steal the students' show, I'm actually going to explain why I think here there's a case. Now, for those who eat egg is about 30% yolk. 30% of that yolk, or more than 30%, is basically repeats. Okay, so what we see when the chicks are incubated, the last few days before they hatch, they actually have prevalence of consuming long-chain fatty acid, in particular DHA and those kind of functional fatty acid. So then the question is, if we pack more DHA or omega-3 fatty acid in these eggs when the breeder are laying them, what will that mean to these chicks? Are they going to have better development of the brain and therefore their behavior, better development of the gut, the immune system, the skeletal system, and the student will explain that. That's what they are basically doing. The other molecule which I look into, and I think also Dr. Mike Steele also look into this, is yeast. Yeast is a very unique cells, and in particular the yeast cell wall, because the yeast cell wall, they are not stimulate the immune system, and as a result produce antibodies. And when it comes to the chicken, when the hen is laying an egg to actually for hatching, she actually deposits a lot of antibodies in that egg to help that chick to be able to overcome the stress she will encounter or he will encounter the first few days post-hatch before they can actually develop the immune system. So then the big question is that if you this broiler breeder these yeast metabolites, do you increase the antibodies in these hatching eggs? In particular, IgA. IgA is very critical for protection of the gut. And these are the questions we are asking. Do you increase the hatching egg antibodies? Do you actually also influence the microbial load in these hatching eggs? Do you influence the progeny performance, and as well as how they deal with the gut health and function? Research is organized in two focus. One is in broiler breeders here, and pullet breeders. So come to broiler breeders, you have the fatty acids being investigated in many aspects, including the behavior. That's why I have my colleague, Dr. Tina Widuski, actually looking at the aspect of feeding broiler breeders, omega-3, and how that influences the progeny, brain development and behavior. We're also looking at many aspects. I don't want to steal the student's show. And now I'll just ask the student to jump in and actually explain further what they're actually doing. And we start with Reza. Reza, stop sharing.
Yeah.
And share. Good.
Okay. Hello, everybody. I'm gonna share my screen. Can you see?
Yes.
Okay.
Yes. Yeah.
So today I'm gonna talk about the effect of embryonic exposure to omega-3 fatty acid on skeletal development in Isobaron and shaver white hens at 42 weeks of age. And today is a part of my PhD project. So in egg industry, we are trying to, we are trying to extend the lay cycle. And the current target is a long life layer capable of producing 500 eggs in 100 weeks. Decrease in eggshell quality and lay percentage are the main reasons for replacing laying flocs around 75 to 80 week of age. So we are trying to have a healthy pullet and turn it to a productive and healthy hen capable of laying for 20 more weeks than what we already have. Around 60% of eggshell calcium comes from diet and the rest comes from a skeletal system. This part is cortical part, which gives the strength to the bone, and this part is the medullary cavity, which it's empty right now at the pullet phase. After the surge of estrogen at sexual maturity, osteoblasts or bone forming cells, they switch from forming cortical bone to forming medullary bone. What is medullary bone? Medullary bone acts as a source of calcium for eggshell formation when the bird does not have access to calcium or diet, like during the dark period. But is it the only part of the bone that can supply calcium for eggshell formation? No. Cortical part also supply calcium for eggshell formation because osteoclasts or bone-eating cells, they are not specialized to uptake mineral only from medullary part. They keep uptaking mineral from cortical part as well. So that means we gonna lose bone strength or we're gonna lose cortical part during the lay cycle. So as you can see here, cortical bone development stops at the sexual maturity or onset of lay. So whatever we want to do to optimize cortical bone has to be done during pullet phase. And then for medullary bone development starts at the end of rearing phase and continues until the end of the lay cycle. So because after sexual maturity, there is no cortical bone formation, we have to maximize or optimize cortical bone formation before the sexual maturity or pullet phase. So Elijah talked about the role of omega-3 fatty acid and some mechanisms of action. So I'm not gonna go through them again. So to date, most investigations on the effect of omega-3 fatty acid on skeletal development focused after the sexual maturity or when there is a high risk of osteoporosis, and there is no information on the effect of embryonic exposure to omega-3 on skeletal development. So the overall hypothesis of my study was that omega-3 PUFA exposure will stimulate the skeletal development and subsequently impact bone and eggshell quality in Isobaron and shaver white hens. And the overall objective was to investigate the effect of embryonic exposure to omega-3 PUFA on embryonic and rearing skeletal development and subsequently impact production performance, eggshell quality, and bone quality in isobrown and shaver white hens. So we offered 3 dietary treatments to 26-week-old layer breeders, including control, like commercial diet without a supplemental source of omega-3 fatty acid, control plus 1% dried microalgae, or DMA, as a source of docosahexaenoic acid, and the last diet, control plus 2.6% of coextruded full-fat flaxseed, or triple F, as a source of alpha-linolenic acid. And the tested diets, diet 2 and 3, they were formulated to have equal ratio of omega-6 to omega-3 and the total omega-3. In my experiment, I had embryonic and pullet exposure to omega-3 PUFA, but today I will be presenting just embryonic exposure effect. So after hatch, all the pullets were fed with commercial diet up to 42 weeks of age. We did some samplings at different ages, such as fatty acid profiling, hatchery performance, body composition, lipid oxidation, indicators, some immune system, bone quality, and production performance and eggshell quality in layers. So result, I will be dividing my result into 3 phases: hatchery period, pullet phase, and laying phase. So before we set the eggs or hatchery period, we did not see any effect on egg component and composition except fatty acid. Fatty acid profiling confirmed the deposition of omega-3 PUFA in eggs by feeding the sources of omega-3 fatty acid. After hatch, we ran DEXA on the body of hatchlings, and the result showed body composition of hatchlings was affected by the treatment. For example, hatchlings from control diet, they had more bone mineral content compared to alpha-linolenic acid group. Or in shaver white pullets, hatchlings from alpha-linolenic acid group, they had higher fat percentage and a lower lean percentage compared with control group. At 18 weeks of age, at sexual maturity, tibia breaking strength and femur rash content, they were increased in shaver white pullets from docosahexaenoic acid group compared with control group. And if you look at the shaver white groups, you can see the interaction between a strain and breeder diet improved cortical ash content in tibia and femur of pullets from docosahexaenoic acid group compared with control group. At 42 weeks of age, at the end of the experiment, there was no effect on bone strength, production performance, or eggshell quality. Tibia medullary ash content and percentage were higher in the docosahexaenoic acid group compared with the rest of the treatments. Previously, we observed bone turnover metabolism, or let's say bone resorption, in broilers was increased when we challenged the birds with Eimeria, Coxey challenge. So for layers, we tried LPS challenge to increase the bone resorption and see how it would affect our parameters. Although production performance, eggshell quality, and bone quality were influenced by LPS challenge, omega-3 PUFA did not offset the adverse impact of LPS challenge in any measured parameters. And in conclusion, breeder feeding with omega-3 PUFA increased fat deposition and decreased lean mass percentage and bone mineral content in the body of hatchlings. Just In shaver white pullets, tibia breaking strength and ash content in the cortical part of tibia and femur were increased in pullets from the docosahexaenoic acid group compared with control group. And at the end of the experiment, egg production, bone strength, eggshell quality traits, they were not affected by embryonic exposure to omega-3 fatty acid. And that was all about from my part, and thank you so much.
Okay. Thanks, Reza. So I guess we're going to have the next student, Rosemary, go, and then we'll keep the questions to the end. Okay, thank you.
You're welcome.
Yeah.
Hello, everyone. Let me share my screen.
Can you see it? Good.
Okay. Can you see the presentation or presenter view?
Good. It's good.
Okay, perfect. So, hello, everyone. My name's Rosie Whittle. I'm a PhD candidate in the Poultry Welfare and Behaviour Lab. This is run by Dr. Tina Wadowski, and I'm doing collaboration with Dr. Kiarie's lab, but looking at more behavioural aspects of nutrition. So my project looked at the effects of maternal omega-3s on offspring behaviour. And in my study, I look at both meat and egg-laying chickens. So I'm just going to run you through what I'm going to talk about very quickly. So I'm going to discuss how omega-3s can affect the brain, go on to explain a little bit about how omega-3s affect humans, move on to animals, and then discuss how behavioural changes can implicate chicken welfare. I'm going to give a brief overview of my research and a summary of some of my current findings. So we've had a general introduction to omega-3s, but I want to give a bit of background about how they can influence the brain. During embryonic development, omega-3s are absorbed by the developing embryo, either from a deposited nutrient source such as an egg yolk or via the placenta. These are the only sources of omega-3s to the developing embryos and must come from the mothers. So omega-3s accumulate in various parts of the body, but predominantly they accumulate in the brain. Many brain functions are actually dependent on omega-3s, including neurogenesis, so the creation of new neurons, dendrite expansion, and synaptogenesis. Research into the effect of omega-3s, particularly on deficiency, on the brain's physiology has found a decrease in the number of neuron cells and the size of them, specifically in regions of the brain such as the hippocampus, hypothalamus, and parietal cortex, as well as just an overall decrease in creation of new neurons. So if we take one region of the brain specifically, I'm going to focus on the hippocampus. The hippocampus plays an important role in both cognition and emotion. Cognition is defined as the mental action or process of acquiring knowledge and understanding through that thought, experience, and the senses. And emotion as instinctive or intuitive feeling as distinguished from reasoning or knowledge. Now, if we know that omega-3s are vital to develop key regions of the brain that influence cognition and emotion, so what we learn and how we feel, we can potentially begin to see how omega-3s could affect behavior. So I'm going to start with talking about humans because that's what we can probably most relate to when it comes to benefits of omega-3s. So Justin Carr and colleagues at the University of Western Oregon created a review which looked at the health benefits of omega-3 rich diets. They found that it could decrease the cardiovascular disease morbidity, decrease carcinogenesis, decrease the risk of Alzheimer's and dementia, reduce the severity of ADHD, as well as hyperactivity and stereotypy in autistic children, decreases anxiety, aggression, and depression, reducing the symptoms of schizophrenia and psychosis, and increasing recovery of traumatic brain injuries. So as you can see, this is a huge amounts of benefits that they can have to our health. But what can they do for animals, specifically animal behavior? So moving on from humans, what do we currently know about how omega-3s influence behavior in animals? So research to date has found both positive and negative effects of omega-3s. These examples specifically cover the maternal feeding of omega-3s, as in my research. So the positive effects so far have been found to increase cognitive ability. So they increase the spatial and working memory. This has been found in rats. Also, they've been shown to increase learned avoidance, or the speed at which an animal learns what to ingest and what to avoid. These have not only been found in rats, but also in red-legged partridges. They are a galliform cousin of the domestic chicken. Another positive effect of maternal feeding of omega-3s, at least in the case of Muscovy ducks, is that they found that they decreased the rate of stereotypic feather pecking. So I've covered some of the positives. However, research has also discovered some negatives. It's been found to increase fearfulness in chicken, resulting in a longer time to recover from tonic immobility. So this is the chicken's play dead response to being caught by a predator. Maternal omega-3s have also been shown to increase neophobia, so the fear of new objects or new foods or new environments. So what could these cognitive and emotional changes have or do to the commercial chicken? How can it affect them? So if we're producing what we could say as smarter chickens, this could be an advantage to farmers. It could increase the speed at which chickens learn to adapt to their environment. For example, a day-old chick which learns faster the location of food and water is more likely to survive. Or a laying hen who learns to lay eggs in elevated nest boxes won't lay them on the floor, which means the farmers can't sell them. Also, improved spatial cognition could help chickens better navigate a complex housing system such as aviaries, leading to less falls from the system or collisions with their environment, which could lead to injuries. However, feeding omega-3s to chickens may not be without their caveats. Omega-3s are also known to increase fearfulness, increasing the fear of novel food environments and objects. This could lead to increased collisions with their environment due to being startled. This could escalate further into sort of smothering events, where the birds are startled into a tight space, which can result in mortalities. Also, neophobia, especially fear of a new environment, might increase the time it takes chickens to adapt to a new environment. For example, when laying hens move from their pullet rearing systems into a mismatched adult housing. This may lead to a decreased consumption of food and water, which can impact or delay production. And finally, fearfulness in itself is considered a negative affective state and may contribute to a decreased overall welfare of an animal. This is why my research is specifically looking at the behavioral implications of maternal fed omega-3s to try and see if there is in fact a balance between increased cognitive ability and increased fearfulness. So far, in my research, I've raised a flock of Ross 708 broiler breeders, which produced approximately 1,000 offspring in 4 different maternal omega-3 combinations. They were either fed a maternal control diet or omega-3 rearing or laying diet. So in this study, I used flaxseed, as Reza previously described. These broiler offspring were tested over 6 weeks in a series of cognitive and fearfulness tests, as well as also doing assessments of leg health. Cognitive tests included a passive avoidance learning task and a team-based test. Fearfulness tests included social isolation tests, a novel object test, and restraint test. Currently, I'm raising 2 cohorts of laying hen pullets. These are Shaver White and Issa Browns, approximately 800 in total, which will be tested similarly to the broilers, but over a longer period of time of 36 weeks. I'll also be adding in a more complex cognitive discrimination task and a human approach test. While most of this has yet to be analysed, I am able to present a few snapshots of my results so far for you. So firstly, I'm going to talk to you about the social isolation test. This involves removing a chick from their home environment and placing them in complete audio and visual isolation from other chicks. This is something that chicks find a little bit stressful because they are very sociable creatures and don't like to be placed on their own. So we place them in a box as on the slide for 5 minutes and record their behavioral response. We look mainly at escape attempts, vocalizations, and freezing behaviors. This test was originally validated in a pharmacological model for anxiety conducted by Kenneth Suthka. So when I ran my analysis, I found that when looking at the effects of the maternal laying diet, so when we fed the mothers omega-3s or controlled diet from 16 weeks of age onwards, the males from hens fed the omega-3 diet vocalized more frequently than all other sex maternal laying diet combinations. This suggests that the maternal omega-3s made the males more fearful or anxious when separated from their flock. And here's another result for you with a little animated GIF to help explain the test for you. So this is a T-Maze test. This looks at spatial memory. So how well do chickens remember the locations of a reward? In this test, we use a mirror as a social reward. A social reinstatement is very powerful. As I've already said, the chickens are very sociable. This is also powerful because they don't see themselves in the mirror. They think it's another chick. So it's a good way of cheating without noise of other chicks influencing their decisions. So there are two different phases to this test. Habituation, so that's getting them used to the environment. As by the diagrammatic, there are 3 mirrors placed in this test, 1 in each arm and 1 at the end of the T. And then for the training and testing phase, the mirror is placed in just 1 arm. So the chickens are required to remember which arm that the mirror is. And the more successful they are in remembering, we can say the more cognitive ability that they have. So this test showed yet another sex and maternal diet effect. But this time it showed that females from mothers fed omega-3 laying diets were the most successful at finding the mirror reward. And both males and females from the omega-3 mothers were more successful than their control maternal diet counterparts. So in just these first 2 tests I presented, I've shown that boys get scared and girls are clever. Well, in this case at least. In summary, maternal fed omega-3s appear to be having sex-dependent effects on behavior, but this isn't a new concept. When looking at prenatal effects on behavior, there's quite often different results depending on the sex of the animals involved. So it's very interesting, but not entirely new. So thank you for listening. I just want to acknowledge the funding bodies that make this research possible, as well as Dr. Kiarie and Dr. Wadowski, my small army of dedicated helpers, and thank you everybody for attending.
Okay, thank you, Rosie. Good. So Ash, are you ready to show?
I am. Okay. All right. Can we all see that?
Sure. Yep.
Okay, perfect. So my portion of this presentation will look at the work that I'm doing for my PhD thesis. So I'm investigating the impact of enriching broiler breeder diets with omega-3 fatty acids on breeders, and then the subsequent effect on progeny performance, immune status, and livability. So just some introduction. I won't spend too much time on it. I know we don't have a ton of time. So broiler breeders and what makes them unique and the challenges of feeding them. So broiler breeders are the parental stock of meat birds, so broilers. And managing broiler breeders to meet the various objectives during the different life stages is a fairly difficult task because you're trying to aim for flock uniformity. We're concerned about egg quality, hatchability of chicks, but these breeders carry the genetics in broilers that for growth and weight gain. So to avoid reproductive issues, broiler breeders are restricted fed in order to avoid issues associated with overweight birds. So restricted feeding in general negatively affects the gut microflora, increases stress levels, and subsequently the potential for disease challenge, affecting performance negatively. Now, ultimately, as the developing embryo is dependent on maternal factors and nutrients deposited in the eggs, there's a potential for reduced chick quality. So that's where this idea of maternal nutrition comes in, where maternal nutrition contributes to the hatchability, which is important because hatching egg producers, of course, are paid based on chick viability, and then the long-term growth and health performance of the progeny. Like Elijah said, the nutrients deposited into the egg are the only source of nutrients available to the developing embryo, and so we're particularly interested in the egg fat components of the egg yolk. They act as a source of energy and essential fatty acids during embryogenesis. So again, I won't spend too much time on this, but essentially we're looking at enriching the diets of omega-3 fatty acids for potential beneficial effects on the chick's long-term health and performance, while also benefiting the broiler breeder. In terms of my objectives, there were three main objectives in my thesis. So first was to investigate the effects of feeding omega-3 fatty acids to broiler breeders during the rearing phase and see how that affected body weight uniformity and their bone quality. We also wanted to investigate the effects of feeding omega-3 fatty acids to broiler breeders during the laying phase and how that affected egg quality, egg performance, and chick hatchability. And then our third objective was to investigate the transgenerational effect of feeding omega-3 polyunsaturated fatty acids to broiler breeders and progeny on progeny growth, feed conversion, breast ear quality, so really production parameters and their response to an enteric immune challenge. So for my experimental design, all the diets for all the phases of the experiment were formulated to meet nutritional requirements. And then the two experimental designs, so algae and LinPro, where I'll refer to algae as DMA and LinPro as full-fat flax seeds, were formulated such that the total ratio of omega-3 to omega-6 remained constant. So even though we were supplying different forms, the ratio remained the same. And the dried microalgae diets were formulated first based on a 1% inclusion level from previous research, and then the LinPro inclusion was determined based off of the total omega-3s in the DMA diets. In terms of my experimental design, so the first column indicates the rearing phase of the broiler breeders. So we fed one of three diets, so the control, the DMA, or the full-fat flax. We then split each diet in the laying phase to be either control and one of the omega-3 sources. And the idea behind that was really to pinpoint where we were influencing the most, and potentially if we didn't see effect in the rearing phase, that could be a cost saving in production. So that was the idea between splitting phases. And then the last column indicates the progeny diet. So again, the control diet was split into control and then either source of omega-3, and that pattern repeated for all of them. In terms of sampling, so we hatched at 3 different ages, so week 34, 44, 54, and 64, and we did that because we know that as broiler breeders age, we see chick quality and production go down. So we wanted to follow that aspect of production. We didn't manage to hatch at week 64. Unfortunately, there weren't enough eggs and production had dropped too much. And then during the progeny phase, So this was repeated 3 times, like I said, at week 34, 44, and 54 weeks. We challenged the birds with Imeria at day 10, and there was a high dose and a low dose. And on day 15, we sampled the 2 high dose birds for lesion scores, organ weights, and plasma, and this was to parameterize the immune response in the birds based off of the inclusion of omega-3 fatty acids. And then we reared the rest of the birds until day 42, so common production age, and the idea of that was to see the recovery from the low dose and how that worked. In terms of results, I will preface this with a lot of these are preliminary results, and there are pieces that I think need to be added in to fully explain what we see. But in general, during the rearing phase of the broiler breeders, we saw a large increase in variation of body weight uniformity in the DMA-fed birds between week 12 to 24, whereas the full-fat flaxseed birds seems to stay relatively consistent in terms of their body weight uniformity throughout their entire rearing period. And then if we actually look at their body weight and body weight gain during their rearing period, there was no interaction effect between the age, which we define by phase, and dietary treatment on body weight or body weight gain. We did see that the DMA-fed birds had the highest body weight gain relative to control, but it wasn't significantly different from full-fat flaxseed. And then the age effect was that body weight increased with age, but this is expected, and the body weight gain was the highest during the grower phase. And so that was the performance data from the rearing phase of broiler breeders. And going into some preliminary results from the hatches. So in terms of average breast weight, our initial analysis showed that there was a treatment effect, a hatch effect, and a sex effect on the breast weight. However, there weren't any interaction effects. So then we wanted to go in and quantify the phase of feeding in terms of treatment, which affected breast weight. So again, coming back to this idea of, is there an optimal time to feed these omega-3 fatty acids? So we ultimately saw that just the progeny diet affected breast weight in these birds, kind of implying that we don't necessarily need to feed the mothers to affect breast weight. We saw that the breast weight increased with hatch weight. So as the broilers got older, so the chicks were heavier and the breast weight was also increased. In the progeny diet, the full-fat flaxseed had the highest breast weight, and the dried microalgae actually had the lowest breast weight. And again, we don't necessarily know how to explain these differences right now, considering these are both sources of omega-3 fatty acids. And then we looked at the average JEJUNAL lesion score. So this was in response to the coccidial challenge we provided in each hatch. And again, our initial analysis showed that treatment hatch and hatch treatment interaction were significant. So we went in again and wanted to see where these interactions were happening. And it was occurring between the hatch and the broiler breeder rearing diet. So in younger broiler breeders, the lesion score in the progeny was highest for algae-fed birds and lower in full-fat flaxseed-fed birds. However, when we get to the later stages of the broiler breeders' production, the full-fat flaxseed response to an enteric challenge is actually worse. So again, something that we're trying to explain with why we see these changes as these breeders get older. And then there was also an effect of laying diet that wasn't interacting with hatch. So as during the laying phase, birds fed the dry microalgae, again, had the highest lesion scores. So again, a poor response to the coccidial challenge. And the full fat flaxseed birds had the best response to the coccidial challenge. But I think some of the samples we've collected for IgA and immune responses will let us kind of explain why we are seeing this. So a lot of this data is very preliminary. And that's all I have. I was like, if anyone has any questions, that's my email.
Okay, thank you, Ash. So we'll continue here before we, start working on the questions, John. So I just want to show you some little bit of data we have done with the broiler breeders, feeding them yeast components. And what we see is that we see increased, IgA in the eggs for hatching eggs. And when we do further investigation, we see that obviously we don't impact the fertility. Fertility, that means the number of eggs with viable embryo after 17, 18 days of incubation. We don't see any effect of feeding yeast on that aspect. We also don't see any effect of feeding yeast on hatch body weight of the chick. You remember the chick body weight at hatch is very important indicator of how they'll perform in the barn. But what we do observe is actually reduced chick variability, which may be important to a hatchery because the more uniform chicks you have, there's more likelihood that you're going to have better performing chicks in the barn. So our work in the East is just new. This is just some small research we have done. In the next 4 or 5 years, we will have students recruited to actually look into this in more details, and we will be happy to share with you in future. Before I wrap up here and open up for questions, this amount of work is not possible without the support we receive. Some of this support already the students have acknowledged. And of course, the many young people or students and research postdoc and technician who actually make this possible. There was a lab in 2017, and I think it's somewhere near the our department, the lobby. And this is a new improved lobby. Thank Jim, look very nice. So, and this is a picture we took in 2020 when we have social distancing. You can already see that. When it comes to collaboration, I really want to thank my colleagues, John Butter from the UofG Pathobiology. He is a well-renowned parasitologist, has helped me a lot establish the Cox Challenge model. Dr. Tina Widowski, we have done a lot, and I continue to learn a lot from her wealth of experience in welfare, and I think there's a lot of interaction between nutrition and behavior and welfare. Dr. Neil Carroll, immunology. I always cherish, talking to Neil on many aspects. My good friend, Dr. Gregory Bedakarat, the poultry physiologist, spent a lot of time, talking about laying hens and, reproduction and nutrition. And finally, Dr. David Ma from Human Nutrition, an expert in fatty acid nutrition. Many funding agencies from ESAC and the industry, we really appreciate the role you play to advance science, to train the next generation of poultry nutritionists. Some of them have already joined your ranks to help the industry move forward. The reality is that the number of people are increasing in this world, and more people want to eat chicken. So we need to have more poultry scientists out there. So we really thank your support. And I think, John, at this point, I'll just see whether there are a couple of questions we can address.
All right. Thank you very much, Elijah, and your co-presenters, Reza and Rosie and Ash. So thanks for showing that to us. Very interesting talk and results.
Yeah.
So in terms of it's very hard to applaud on on this distance. But if there are questions, then you can either just unmute yourself and ask a question, or you can write it in on the conversation on the side, and we can translate those questions to the group. So I'll just leave it open here for a sec while people organize their thoughts. Or maybe I can ask a question to start things off.
Yep, sure.
You show those effects of omega-3s, you know, in the maternal diet, let's say, and, you know, how it affected things like behavior. And maybe that's difficult to kind of postulate mechanisms of what's going on there because of the
yeah.
What about the body composition stuff? So about the changes in the body fat and even the skeleton mineralization, like what do you think is going on there?
I think there's, I can speak a little bit to it, and then someone like Reza actually did much more deeper analysis in this. I think there's repetitioning. We see, we first of all start changing the embryonic stage, the utilization of certain nutrients. We see some shifts in there, which actually end up affecting, you know, the body composition. We actually have a lot of this data, John, where we have used DEXA to actually see the changes we are making. We didn't have time to share all this stuff, but sure. Some of the data is surprising. I think, Lesa, do you have some comment to say what you found in particular laying hens?
Yeah, sure. So as Elijah mentioned, we did DEXA on the body of hatchlings, and what we saw, embryonic exposure to omega-3 increased the fat deposition and decreased the lean and bone mineral content, which was not something that we were looking for, right?
Yeah.
But if we just talk about mineral content, yes, we were not looking for that, but we measured also collagen type 2 and collagen type 3. So they are the foundation for mineralization of a skeletal system, right? So we observed improvement in the concentration of collagens in a skeletal system. So I think this story has different sides, but in terms of body composition, yes, the effect was significant, and I believe it comes from embryonic uptake of nutrients, because we measured the uptake of nutrients during the hatchery period, and we observed when we have omega-3 in the egg, we have less uptake of dry matter, protein, and mineral by embryo. So this is something we need to consider when we are using breeder feeding technique, because we want to have a smaller yolk sac attached, right? So when we are adding omega-3 in the environment, in the egg, we're going to have a little bit bigger yolk sac, which increases the chance of diseases, yolk sac infection. So I cannot say it was a positive effect or negative effect, but it was really complicated. But in terms of body composition, the significant part was less mineral content, more fat, and less lean in the body of hatchlings.
Okay, good. Right. Let's see. Thanks, Lesa.
Yeah, thank you. Any other questions?
I think Tina had lifted her hand.
Yeah, I have a question for Ash, actually, because Ash, you're seeing a lot of hatch effects, and that you said you can't explain. And I'm wondering, we've been doing a lot of work looking at the effect of maternal age on stress physiology and behavior. And there's a lot of egg composition changes that happen as the hen gets older, fatty acid composition, as well as steroid hormone, as well as yolk to albumin ratios change significantly, which we think are likely responsible for some of the behavioral differences depending on the mom's age. So did you look at any just changes in egg composition over time?
Yeah, so those are parameters that I've collected and haven't run the statistics on. So I think that will definitely explain some of the change that we see. So I looked at the changes in the actual fatty acid composition within the yolk, and then looked at the composition changes of the egg itself. So the albumin weight, the hoof height, and then the yolk weight, and looked at how that changed over time. So I do think that would be a factor that would explain some of the changes that we see in terms of the hatch effects. And then we also looked at breeder performance in terms of body weight uniformity all throughout the entire production period, which again is something that I think would affect the production and the results that we see.
Yeah, it's really interesting because nobody's really looked at the age of the breeder flock effects anymore, and they're hugely different whether you're using young or old hens. So,
yeah, especially even if we think of something like yolk sac infection, right? We generally see that in older birds because they're more susceptible to the bacteria. So yeah, no, very cool. I'm very excited to continue to kind of tease out why we see these effects.
Good, thanks.
Thanks, Tina.
It's a question from Christoph.
Yeah. Oh.
Are you going to make me unmute here? I'm sorry.
Yep.
Yeah.
No
anyway,
worries,
thanks for the presentation. So as per the what I've said in the chat, I guess my question is, basically, if we're not having to starve heavy birds, broiler breeders in particular, does this free us up to deliver more of the nutrients you're talking about today? Like, so do lower growth potential genetics actually offer us more opportunity to do the things you're talking about today?
Yeah, so Christoph, this raw broiler, I think my colleague, Dr. Widowski, we are doing some more work on these raw growing broilers. But currently, the way we manage the broiler chicken we have, the broiler breeders we have to restrict feed them. So just because we don't want them becoming fat, or because when it becomes too heavy, they eat too much, you actually affect their reproductive performance. And the broiler breeder farmers are paid by the number of hatching eggs or they produce over a lifetime of that broiler breeder. So we limit the feed, but I don't think the question you're trying to address here is a question of feed restriction. No, we are actually trying to address whether feeding omega-3 or yeast, we actually enrich these eggs further. But it's also important to consider these hens are actually more hungry to meet their energy needs. So you actually may have less being allocated to the eggs, but that's something you have not looked at as yet. So, yeah. Ming?
Yeah, Elijah, yeah, a very interesting presentation by your group. I'm working with Tina's group. I heard about mentioning the maternal manipulation in poultry here. I was wondering, being reported now, even the newborn mammals now being showing the gut. A newborn animal have microbes even before birth. Yeah. I wonder in the poultry you might have advantage that inject maybe probiotic bacillus into the yolk in the gut, so be colonize the gut with healthy probiotic bacteria in a way, in terms of manipulating maternal manipulation, right? In terms of yolk composition or probiotic bacteria there. So we're early on manipulate the microbiome poultry in a way. That could be the interest. I'm not sure what has been done there, in terms of apart from nutrient, but microbiome early on hatching process. Any comment on that?
Yeah, thank you, Ming. That's a very good point. As I said, there's a lot of interest of novel application of nutrients as well as additives like probiotics, and there's actually a lot of work showing there's some potential of actually putting probiotics in the eggs before they are hatched. The challenge we have right now is that this is as good as academic research. Still at the commercialization level, the novel feeding is not practiced for many reasons. Some of them has to do with the fact that someone has a patent over that, and also I think also the practical aspect of it. I haven't found a hatchery person to explain to me why the hatchery has not picked up these kind of approaches, but there's a huge potential to do this with chicken.
Thank you.
So we may have gotten to the end of questions, Elijah.
Yep.
I see a hand is up here again.
Let's address it.
Maybe that's an old one.
Yeah.
Yeah, thank you very much for that talk. Got us thinking about different things, and to me, it's very interesting, this whole connection between nutrition and behavior. That will be interesting to see how that plays out over the next decade or so, and the kinds of things that are found out there. Plus all the other collaborations you have going on and various things. And I was going to list all the different types of nutrients that I see you got papers, publications on, and it's a very long list of different factors and sources that you're working with. So I'm sure lots of very cool, interesting things are going to keep coming out from your group. So thank you very much for that presentation. Yep. And I hope everybody enjoyed, and we recorded the session so that we will be able to put it up on the CNM website at some point in the near future. And so on behalf of the audience, thank you very much.
Welcome. Bye-bye, everyone, and see you in the future.